Vision. Part 2. Physiology of the Eyes. Based on the Inner World, Beyond the Prism of Senses by Mohammad Babaee. This section of class is about physiology of eyes. If you remember, in the past sessions, we talked about how and where to start our journey of self-awareness. We mentioned that our integrity, beliefs, knowledge, and memories are all based on the information that comes to us through the five senses. In that case, the five senses are our only source of information. So, if the foundation of all these beliefs is these five senses, let's examine and see if they provide us with accurate inputs. The focus of today's lesson is exploring the physiology of the eyes. Let's begin with the sense of sight, because vision is one of the most important and reliable human senses and shapes the foundation of many of our beliefs. Our question is, how does the eye provide information to us? The eye is the first region where the process of vision takes place. We want to investigate the accuracy of the image formed via the eyes and whether the visual channel can serve as evidence for the existence or non-existence of something. We can see, and the process of vision has become such a natural phenomenon for us that we might not even think about it. We rarely raise questions about how we see. The information received by the eyes, to what extent does it align with the reality of the world? Are these pieces of information reliable enough for us to form opinions and judgments about the intricacies of the world? To comprehend this matter, we need to investigate how the eye functions and provides us with information. First, light encounters the eye, and this interaction takes place in a region called the retina. On the surface of the retina, there are light-receptive cells known as photoreceptor cells, namely cones and rods, which are sensitive to light. Photoreceptors have ion channels on their surface that undergo structural changes upon exposure to light. Let's delve into the intricacies of these cells and examine what happens within them when exposed to light, and whether these events are related to what we perceive or not. On the retina cell walls, there are ion channels resembling gateways regulating the entry and exit of substances into and out of the cells. These channels act as gatekeepers, controlling what substances are allowed to enter, exit the cells. There are two categories of ion channels on the cell walls. The first category includes the entry channels, determining which substances are permitted to enter the cone and rod cells. The second category comprises exit the channels, responsible for the exit of chemical substances. The molecules entering and exiting the cells, or ions, are particles that carry positive or negative electric charges. Ions enter the cell through these ion channels and exit from it. This entry and exit of ions change the electric charge inside the cells. When the number of positive and negative ions entering and exiting is equal, the charge inside the cell remains unchanged. However, when the balance of ion entry and exit changes, the charge inside the cell also changes. In conditions of darkness where no light reaches the eyes, the equilibrium between ion entry and exit is maintained. However, upon entering an illuminated environment where light strikes the retina, the entry ion channels on the cell surface undergo a change in their open-close state. This modification in shape leads to the closure of the entry channel, preventing the entrance of ions into the cell. Concurrently, the exit channel continues expelling positive ions from within the cell, resulting in the internal space of the cell becoming negatively charged. At this point, as the interior of the cell acquires a negative charge, it functions like a charged capacitor. When the negative charge reaches a specific threshold, it discharges from inside the cell in the form of an electric spark. Subsequently, this charge travels through a network of nerves to a specific location at the back of our brain, forming an image. In upcoming sessions, we will delve into the analysis of this image. The next point here is that after the encounter of light with the cellular channels, these channels remain closed, and it takes a fraction of a second for them to reopen and return to their initial state, during which brief return we lose the ability to see. Looking at the brain, we won't see anything called an image. Chemical reactions, the opening and closing of cellular channels, create neural messages, and these reactions form the everyday world we experience. Knowing these facts, let's take another look at the world around us. Light from objects around us reaches the eyes, stops in the eyes, and conveys a neural message to the brain, where an astonishing phenomenon of vision unfolds. Let's summarize key points briefly. When light interacts with the cells of the retina, the alteration of the cell wall structure, which contains entry and exit channels, results in the generation of a neural message that travels from the eye to the brain. The incoming light stimulates the cells of vision, and this stimulation is transmitted to the brain through the neural network. Which part of this process can create an image? Let's imagine we extract a brain and place it in a life-sustaining solution in a laboratory, connecting it to a device which creates electrical signals. Assume that these electrical signals stimulate the nerves that are connected to the parts of the brain that are responsible for vision. What will happen? Remarkably, it will perceive a vivid world within itself, a world complete with a physical body, movement, sight, and the experiences of eating, interacting with friends and enemies, working, and being part of a family. If a figure resembling a teacher appears in the brain's dream and tells it that its experiences of movement, eating, and living are illusions, that it is merely a spectator in a fabricated story and film, how would the person react? Is the external world identical to the one constructed inside our brain? Does the sky we perceive internally truly mirror the external sky? Given that our sole link to the external world is through light that terminate at the eye's lens, how can we determine the answers to these questions? Join us as we delve deeper into these mysteries for a greater understanding of ourselves.